Hello to your 5 students. Today we will be talking about tissue engineering. This is a type of regenerative medicine which involves the development of biomaterials and the practice of using scaffolding, cells, and biologically active molecules to create functional tissues. The main goal of tissue engineering is to assemble, maintain, restore, or improve damaged tissues, or even whole organs. Tissue engineering, being a mere 30-year-old field of research, has the potential to solve many health issues, such as producing artificial skin. Now let's take a look at one of the many procedures that outlines the cycle of tissue engineering from development to use.
Here lies Freddy, a young boy in need of a new liver. One method is to do this in vitro, which means most of the development will take place outside of the body. First, the cells extracted will come from good old Freddy himself. Then the cells from Freddy are expanded upon and manipulated in order to have more biomaterial to work with. This new quantity of cells is combined with mechanical and molecular signal, which are essentially a set of instructions for the cells, into a scaffold, which is an environment to support the cells, which is also known as cell seeding and extracellular matrix expression. Additionally, the construct, the thing that we're making, is then implanted in a growth medium to allow the cells to properly express and grow the organ. Finally, this is incorporated back inside Fred's body for viable use.
Let's apply our tissue engineering diagram to a real-world application. Xenotransplantation is any procedure that involves the transplantation, implementation, or infusion of live cells, tissues, or organs from an animal source into a human recipient. A prime example of this is with pigs. Pig kidneys, livers, and other organs have been examined as possible organ donors for humans. Currently, there has been some success with kidney xenotransplantations. According to the National Kidney Foundation, over 3,000 new patients are added to the kidney transplant waiting list each month, with someone being added to the list every 14 minutes. Researchers are looking for a solution and are turning to pig kidneys as a substitute.
Genetically engineered pig, a kidney becomes more viable to transplant into humans. Specific genes are removed from the pig, and others are added through the process of deletion and insertion. This makes the kidney suitable for the human body and prevents hyperacute xenograft rejection, which is a fancy way of saying incompatibility between the recipient and the organ. The pigs are raised on a pathogen-free facility and are selected through tissue matching to the patient. From here, the kidney can be surgically removed from the pig and is now ready to be transported to the human recipient. Pig kidneys are chosen since they have a similar blood flow to humans, as well as balanced fluids and electrolytes. At this point, the process is very similar to a normal transplantation. With the help of tissue engineering, xenotransplantation is becoming a promising method of saving lives. At the University of Alabama at Birmingham, this procedure was made a success. Jim Parsons, a 57-year-old brain-dead recipient, received 2 genetically modified pig kidneys. While on a ventilator, the kidneys function properly. This has been a major breakthrough for xenotransplantation. However, challenges still arise, such as infection, lifespan, and overall health of a human with a foreign organ.
Pig kidneys are just one example of how tissue engineering is paving a way for new discoveries and innovation. This field is versatile and represents potential alternatives to contemporary solutions.